Today we are largely talking about some of the most critical techniques in immunology, and with one exception, they are all techniques that involve the use of antibodies. So there are some other critical things in immunology, but these are the antibody ones. Many of them, as you will note, are things that are not necessarily immunology specific. So they are used in cell biology. They're used in a huge number of different types of processes and subdisciplines. And so even if you never do immunology again, you will probably use some of the things we talk about today because there are so many techniques that involve the use of antibodies. The first thing that we need to talk a little bit about is understanding the difference between polyclonal and monoclonal antibodies and how we get each of them. Because most of the techniques that I'm going to tell you about use monoclonal antibodies. It's not 100%. You certainly can use polyclonals for some of them, but very often we use monoclonals, and so we need to talk a little bit about monoclonals and where they come from. So if you recall, a polyclonal set of antibodies are antibodies that all bind the same antigen, but that bind multiple different epitopes. So here you can see there's a mixture of antibodies, the red one, the blue one, and the black one. They all bind to myoglobin, but they bind to different epitopes on myoglobin, either the red triangle, the blue square, or the gray thing. Alternatively, and that's what happens if you actually look at an antibody response in an animal. You get a polyclonal response. Often when we are doing the techniques that we're using, we want to use monoclonal antibodies. And with monoclonal antibodies, you are going to have purified populations where you have antibodies that are all recognizing the same epitope on that antigen. As I mentioned, you get polyclonals when you are doing an actual response in an organism. And so in order to obtain a monoclonal, you need to do some additional work in the lab. Yep, Mark. Basically, yeah. So with a monoclonal, you're going to get a much more sort of standardized reaction. Our ability to make monoclonals and polyclonals also is somewhat related to something that has come up in lab thus far, which is that you've seen a little bit about differences between primary cells and cell lines, and we are going to talk about that considerably more as we go forward. Primary cells are cells that come right out of an organism, like the cells you took out of the mouse the other day. Cell lines are cells that are immortal for some reason. They have some type of alteration, so they will divide indefinitely in culture. And the THP1 cells that you are using in your experiment that you've just set up an experiment plan for are an example of this. The awesome thing about cell lines is that they grow indefinitely in culture. If you need some THP1 cells, you can go in the freezer and get some, and you'll have some today and some tomorrow and some the next day. And if you need a huge number of them, you just let them grow for a while till you get a bunch and you're all good. But they had to be modified in some way in order to become immortal and to undergo infinite cell divisions in culture. Primary cells came straight out of the organism, so they're pretty realistic, they're pretty physiologic, but they probably won't divide for very long before they die. And so if you wanted to do an experiment this week with some primary cells, you'd have to, get a spleen from a mouse and do that whole prep. And then if you wanted to do that same experiment next week, you'd have to get another spleen from another mouse and do the whole prep. And you'd have to keep doing it over and over again. When I was a graduate student, sometimes if I really kind of was doing a lot of work with my cells, I would get my primary cells to live for 2 weeks, but they were not in great shape. And so you have a really limited lifespan. If you wanted to get a lot of them, that means a lot of mice and a lot of spleens. And so there sort of are these pros and cons to both of these cell types, and you will see these cell types in the production of monoclonal antibodies. And so if we were to take an antigen with 4 epitopes, shown here, 1, 2, 3, and 4, and inject a mouse with that antigen, our mouse would make a polyclonal antiserum. It would make some antibodies against antigen 1, against antigen 2, 3, and 4. I'm sorry, epitope 1, 2, 3, and 4. And you can see that here. If we isolate the spleen cells from our mouse, we can find a B cell that makes antibody number 1 against epitope number 1. We can find a B cell that makes antibody number 2 against epitope number 2. We can find B cell number 3. We can find B cell number 4. And so you can see those B cells in the test tube. Now, if you think about it, we could pretty easily actually differentiate those B cells, figure out which one bound to which epitope, and then just like sort like the one you wanted. You could just get B cell number 1 and put it in a dish. And then you could leave it in the dish and hope that it secreted antibodies. And they would be monoclonal. They would all be against the epitope that you wanted. But realize that these are spleen cells that you just got out of your mouse. So are these primary cells or are these a cell line? These are primary cells. And so what is going to be your problem in doing that experiment? They'll die. That cell will live for a little while. It might make you some antibodies, but then that cell's going to die. You're not going to make a lot of antibodies. You're not going to be able to have a huge number of that cell making you antibodies. And so maybe, like, for some kind of experiments, that might be enough antibodies, but probably not. And certainly, if you are the antibody company who was going to sell this antibody, that is completely not going to work for you. So the antibody store cannot do that. So instead, what was done, and the people who figured this process out won the Nobel Prize, is that those spleen cells are then fused with a cell line that is immortal. So we use a chemical that will actually fuse together the spleen cell with an immortal cell known as a myeloma cell. And so you will get these fused cells. You can see that there's a cell at the top that's a fusion of the spleen cell number 1 and a myeloma cell. Number 2 is a fusion, number 3, number 4. Because they are fused, because they are hybrid cells, we call this making a hybridoma. And we can grow that hybridoma in special media that kills any unfused cells. And so in the end, the only cells that live are the fused cells, because these are going to live for a short time anyway, so you don't really care. These get killed by this media, and these are the only cells that can live in your special media. And now you've got the benefit of a cell that makes your antibody against your epitope of interest. It has done the VDJ that you want, but it has also gained immortality from being fused with an immortal cell line. So you can then sort out those fused cells and now grow your hybridoma forever and ever and ever and get all the antibodies you want and get all the monoclonals that you want. So what you can see is that there are a few different types of sort of lab manipulations that have to happen in this process. But by and large, if you are doing any of the techniques that I mentioned to you today, which most all of you will have done at least once, you were using monoclonals that were made through this process. that you purchased from some company. Yep.
What do you, like, how do you put that?
So basically, you're using something it's a lipid, approximately. You're putting it in, and it actually makes the cell membranes of the two cells fuse.
So how does it not just make one giant blob of cell?
Careful titration. Yes.
So how does the genetic material of both cells get incorporated into one cell?
These are weird cells. Absolutely weird cells. So yes, they are going to have a lot of genetic material. Yep.
So they have two nuclei.
I don't remember if the nuclei fuse or not, but they have double genetic material, certainly. This is the process that is used for making antibodies for most of our techniques. One thing that I would like you to notice is that if we do this whole process, the antibody that we will get out has a constant region. It'd be really nice if I had colors today, but oh well, I'll survive. The antibody that we get out has a constant region that came from a mouse because it started as a mouse spleen cell. That constant region is encoded by a constant region exon from the mouse genome. And so it has a mouse constant region. If I made this if I had used instead at the beginning a goat instead of a mouse, I would get an antibody that came from a goat spleen cell, and it would have a goat constant region, etc., etc. That is going to become very important later. So now just realize this mouse antibody has the mouse constant region. So these are some of the assays that exist that use antibodies. The thing that I want you to notice about these assays is that one of the big variables that we see is a variable in terms of antibody sensitivity. How much antibody is required for detection and how much antigen is required for detection. So some of them are incredibly sensitive and can detect low levels of antibody or antigen. Others require really high levels. The other big difference that you'll see is the format of the antigen that we are using. The simplest of these and the oldest of these are the ones that are shown at the top that have pretty low sensitivity. They are all assays that function based on this idea of antibody agglutination or antibody precipitation. They are called either precipitin or agglutination reactions. There is a technical difference between the two, but we are not going to worry about that right now. And the idea with any of these antibody precipitation types of experiments is that if you have a lot of antigen and a small amount of antibody, you end up getting the antigen bound to the antibody and whatever. If you have a lot of antigen, or a lot of antibody, a little bit of antigen, you get some binding. Yeah, that's great. If you have just the right amount of antigen and antibody, you can make higher order complexes where multiple antibody molecules are binding to multiple antigens are binding to multiple antibodies, and so you get this big cluster of antibody and antigen molecules bound together. And so you can see that we get this at sort of the middle concentration of antigen and antibody. You will see over the course of this semester, one of the most important concepts in all of immunology is Goldilocks and the three bears. Too much is bad, too little is bad, has to be just right. Too little, too much, just right. When you get this large higher order complex, that complex will precipitate out of solution, and it will make a clump. And so you can look for clump formation, which will tell you if there are antibodies present that bind your antigen, or antigens present that bind your antibodies. If you mix them to get things together and you get clumping, then you can measure that clumping. The most famous way that we do this is to look at carbohydrate antigens that are found on blood cells and to do blood typing. Yep, Mark.
Why does too much antibody decrease?
Because if you have too much antibody, then each antibody is binding to one antigen, and you're not going to have a high probability of each antibody binding to more than one antigen molecule. You're diluting out the antigen, basically. All of your blood cells have carbohydrates as terminal molecules on those cells. Everybody's has this structure, which is known as the O antigen. Some people have an extra GalNAc. Some people have an extra galactose. If you have an extra GalNAc, we call that A. If you have an extra galactose, we call that B. And this results in your blood cells either having absolutely no terminal carbohydrate and being O, 2 copies of A, 2 copies of B, or A and B, depending on what your parents gave you. And so these different red blood cells end up being different antigens because of the carbohydrates that they have on their surface. And so we can mix blood cells with antibodies against either the A or the B carbohydrate and look to see whether we get clumping. So we can do blood typing with one of these simple precipitation reactions. There are also some similar assays where we can look at blood cell clumping based on carbohydrates to diagnose a couple of other conditions. Officially, this is one of those. This isn't a blood typing example, but it gives you the idea. Where in some situations, all of the antigens and antibodies cluster in a little group right at the bottom of the well. And in other cases, they don't. They're sort of spread out and not making that nice clump. And so we can actually examine the differences in antigen-antibody binding. To give you another example of blood typing, this is a blood typing card. It's a piece of paper that has antibodies in 4 different circles. So this one has an antibody that binds to the A carbohydrate. This one has an antibody that binds to the B carbohydrate. This one has an antibody that binds to what's really the RH. And this is a control, a negative control. And what you can do is you can put your blood, just a drop of blood, out of your finger onto this card. I actually did this live in front of the immunology lab one day. I did not know my blood type before that. And what we learned is that I have B positive blood because I got clumping with the B antibody and with the RH, and I did not get clumping with the A or with the negative control. And so that's all blood typing, a quick blood type is one of these types of agglutination reactions. Yep.
So if you had O blood type or O negative, you wouldn't get clumping for anything?
If I had O negative, I would get no clumping for anything. And if this doesn't make a ton of sense to you and you want to further understand it, if you look at the link at the bottom of this slide, the Nobel Prize Foundation actually has a game on their website where you can look at this. And I have it linked on Moodle as well, but the link on Moodle is wrong. This is the right link. And so what happens with this, I'm only going to go partially through it. Where's the game? I want the game. Do you want to reach the game? Play, yes. Play the game. Oh yeah, see, there was a car accident, and we're going to skip the intro because basically you're trying to find a blood transfusion for the people. Yes, the people need blood. We got it, thanks. Start now. All right, so we have our person. We're going to get some blood from him, I think, if I remember how. Hello? Hello? Perhaps you should read the instructions before you're doing the game. But basically what's going to happen is you're going to get some blood. You're going to put it in the tubes, and you're going to look to see if there's clumping. And then you'll be able to tell which types of blood he can have as a transplant. And so this shows sort of the process, again, of the agglutination assay, even if I can't pull it off right now. Yep.
And this happens like relatively fast.
This happened, I did it, it's within 5 minutes, if that. Yeah, it's an automatic thing. Yep.
Can't you just cheat the game and give him O negative every time?
If you had enough O negative. O negative is not very, you don't have enough O negative to do that. Yes?
So what is the RB? Is it another kind of carbohydrate or is it like a protein?
It's called the RH antigen, and the RH antigen is another carb. There's another very famous example of a precipitation or agglutination assay, which is a pregnancy test. So this is actually how a pregnancy test works as well. So with a pregnancy test, you have it's basically a piece of filter paper. You put urine on the filter paper, and if you're pregnant, that urine contains a specific hormone, HCG. See, it rises in pregnancy. Anyway, you put that urine on the piece of filter paper. If you're pregnant, it has the antigen in it, which are these little dots. It will then move across the filter paper, basically wick across the filter paper, because that's how filter paper works. And the area right next to it will have some antibodies that will get carried further down the filter paper because you've added liquid and now you're getting the liquid wicking further down the filter paper. There are going to be two regions where there are other antibodies. One of them is an antibody against this antigen. If the antigen is present, then you'll get a complex made. You'll get the precipitation happening. And you'll be able to detect that the antibody that you picked up here was actually bound at this location because this antibody is able to bind this antigen and has a color marker on it. You also have another control line where this antibody just binds to this colored antibody that you have that makes a second line. And so it's really just a precipitation reaction again, whether or not you're getting precipitation at these two different types of lines. Sometimes they do fancy things like make the lines perpendicular so you can get a plus or a minus, but it's all just a precipitation reaction looking here for antigen. In the previous place, you were looking for the antigen on the red blood cell. Many of the rapid at-home tests for things tend to work like this and tend to be precipitations. One of the other things that we can use antibodies for is a technique that you have seen in Bio 250. There are actually a few techniques you've seen in Bio 250. And so one thing that you could do is you could look at slides under a microscope. And you could take your slides under a microscope and you could use antibodies to detect some type of antigen that's in that slide. Perhaps that antibody will have some type of indicator that is attached to its Fc portion that will let you look for your antigen of interest. And then what you might find out is, where in my cell is this antigen? Or if you're using tissue sections, you could find out, where in my tissue is this antigen? Is this antigen in this tissue? You could find out a lot of different things like that. You get a really nice sort of picture. Sometimes you can't buy an antigen against, or an antibody against your antigen of interest that also has an indicator on its Fc portion. One of the things you can do in that situation is that you can use a secondary antibody. And so here you will have an antibody that binds your antigen and a second antibody that binds to the Fc portion of the first antibody. That's called the secondary. And the secondary will have some type of indicator on it. Typically what will happen is you might have something like your antibody against your first antigen be an antibody that was made in mouse. You can then use a secondary that recognizes the mouse constant region. And so you'll end up with some antibody that binds to the mouse constant region. Usually, you do not make this antibody in a mouse because the mouse constant region will be a self-antigen that would give the mouse autoimmunity. That would be bad. And so instead, you make it in a different organism. And so you might make it in a goat. And so then you will end up, this is usually referred to as goat anti-mouse. And this is mouse anti-whatever your other antigen is. And so that tends to be the way you could set this up for your immunostaining. That could be really useful if you have a tissue sample or if you have something that you want to look at in a microscope, but you might not have that. You can use the same principle if you have some cells and you want to look at their contents and you just lyse the cells. So you could take that same set of cells that contains a whole bunch of different proteins and you can lyse them. You also will denature them, and that's important to realize because that is going to change which antigens are available. And then you could run them on a gel, an SDS page gel. SDS page gels, if you remember from Bio 250, are super flimsy, and you spend most of your time being afraid you're going to break it. So it would be hard to spend a lot of time working with that gel, but you just spend all that time separating your proteins by size. And so the next thing that you do is you take some electricity and you transfer the proteins out of the gel onto something solid that doesn't break as easily. And so you transfer it onto this membrane, but happily the proteins stay separated. You can then put antibodies on either a primary that has a detection molecule or a primary and a secondary and get a band that tells you where the protein is. The nice thing here is that you get some additional information about the molecular weight of the protein. Before, you got some information about where in the cell that protein lived. Now, you have some information about the molecular weight of the antigen. And again, you could say, is the protein present or absent? All that good stuff. So it's exactly the same procedure. It's just with a different form of the antigen to start with. You could also imagine that you don't even have that. And so you could do the exact same thing, but with a different format of your antigen, where you might have your antigen in solution. We are going to do this later in the semester to help us measure a cytokine that was produced by a cell. And so in the lab, the antigen will be the cytokine. And we will have a tube of stuff the cell made, of liquid the cell made, and we want to know, is the cytokine in there or not, and how much. And so what we can do is we can take these little plastic wells and put antibodies in them. We can coat them with antibodies. Those antibodies will bind to the antigen of interest. So in our case, it would be the cytokine, but it could be whatever. We'll then add our sample that we hope has the cytokine. If it does, the antigen will bind to the antibody. If not, then nothing will bind to the antibody. We can then use another antibody that can bind to that same antigen, and it will also be trapped there. If there had been no antigen binding, there would be nothing to trap this antibody, this detection antibody, and so it would not stick. There's wash steps between all of these. And then you can detect this either because of something like an enzyme labeled E that can convert a product, or you can have a secondary, just like you had in the other ones as well. And so you can see there was no antigen, so we got no color change because this antibody didn't stick and make our substrate change color. Here, there was definitely antigen because we got a color change. And we can even see different amounts of color change, telling us that there were different amounts of antigen. We can compare to a standard curve. We can get all sorts of useful info. This is called an enzyme-linked immunosorbent assay, or an ELISA. And ELISAs are one of those techniques that immunologists use all the time. There is another way that you can do an ELISA as well. So this ELISA that I am showing you is an ELISA where you are testing for antigen. You are saying, is my antigen present? Technically, this ELISA is called a sandwich ELISA because you are sandwiching the antigen between two different antibodies. You can also set up an ELISA to detect antibody instead of to detect antigen. So you can flip the ELISA around where you can detect antibodies. This is the ELISA where you're detecting antibodies. It's called an indirect ELISA. If you go to the doctor's office and ask for an HIV test, you will have an indirect ELISA performed to do that HIV test. So what they will be doing is they will be looking to see if your body is making a bunch of antibodies against HIV. And it's looking for a bunch. You have to have a lot of antibodies for this to be positive. So the fact that you all currently have one B cell does not matter. It's the fact that you need a lot of them to have enough antibodies for this to be positive. What they will do is they will stick antigen in the plastic well. Before we stuck antibody, here we're going to stick antigen in. And then we're going to put your blood sample. If you have HIV, you'll have antibodies against it, and they will bind to the antigen. If you don't, then nothing will bind to the antigen. And then we will use another antibody that will bind to the constant region of the first set. We will use an anti-human. Perhaps we will use mouse anti-human or goat anti-human or something that just binds to constant region of human antibodies because you are a human, so antibodies that come from you are human antibodies. And that one will have your enzyme, will convert the substrate, will give you a color change. And so you can also detect presence or absence of antibodies with an ELISA, in addition to detecting presence or absence of antigen. There is also a way to do some quantification with this type of ELISA. All right, so these all are sort of things that seem pretty straightforward, things that are sort of related to things you've done before. This is also related to something you've done before. We did a lab in lab 1 where you had to tell the difference between a whole bunch of white blood cells by their morphology under the microscope. If you remember, it was a little tricky. It wasn't super fast. It's not always the easiest thing to do. And if you actually then take into account how rare the white blood cells are, you can see that if you actually really want to give good measurements of white blood cells in blood, life is going to be tough. But there's one additional complication beyond even what we did in the lab 1. So if you think about lab 1, how many of you saw a lymphocyte? Probably most everybody. Was the lymphocyte that you saw a B cell or a T cell? What do you think? No idea. If it was a T cell, was it a CD4 T cell or a CD8 T cell? No idea. And so you can see that you're not actually really able to look at the full heterogeneity of the cells that you're looking at. So if we look in reality, lymphocyte could be a B cell, it could be a T cell, it could be an NK T cell. That T cell could be a helper or a cytotoxic. It could be one of many subtypes, and you can't tell the difference between those at all under the microscope. And so this graph on the right-hand side is showing you, out of all the lymphocytes, what percent are each of these different types of cells. And so you might need to do experiments where you can actually distinguish the different types of these lymphocytes, and microscopy is not cutting it for you. So I have a graph that looks something like this in my PhD dissertation. At some point I'll actually show you the real graph and not just a cartoon version of the graph. Where I was giving mice a virus infection and then measuring the number of CD8 positive T cells they had that were responding to the specific epitope I cared about over time. And so you got to see the primary response and secondary response. So I was measuring this one specific T cell response in those mice. And I measured it lots of different days, and I measured it to make this graph. But what that means is I had a way that I could tell the difference between a CD8 T cell that responded to HIV and a CD8 T cell that responded to the flu in the blood of my mice. And in reality, I had to be able to take blood from my mice and distinguish the CD8 T cell that bound HIV from all of the other cells in the mouse. Yep, Mark.
But if the mice were like HIV infected and not flu infected, wouldn't you be able to assume that there would be a lot more CD8 or like T cells in general? Or like
You would hope that there would be more, but I think on the max day, please note that I defended my dissertation 10 years ago, and I'm trying to remember numbers off a graph that I made more than 10 years ago. I think like the max for the primary response was 4% of the CD8 T cells were against the antigen I wanted. Maybe in the secondary it got up to 20. I really don't remember. So yeah, they're enriched, but it's still not all of them. The way that we deal with this problem is that we have realized that leukocytes have unique proteins on the surface of their cells. So a T cell is unique because it has a T cell receptor. It has a protein called CD3. If it's a CD8 T cell, it has a protein called CD8. Our CD4 T cell has the T cell stuff, T cell receptor and CD3. The CD4 T cell also has one called CD4. There's one that neutrophils have, there's some that monocytes have, there's some that NK cells have, there's some that B cells have. And so what we could, so before I say anything else about this, there was a day at the beginning of the semester where I saw some of you guys after class. And I believe somebody said something to the effect of immunologists are no longer allowed to name things. Because you found like the names of stuff were super weird. And you're like, somebody needs to just like control this and standardize this and make it not so crazy. And immunologists have realized that problem. So this was actually the first paper I had to read in grad school. So this paper says the CC chemokine thymus-derived chemotactic agent for TCA4, secondary lymphoid tissue cytokine, 6C-kine, Exodus 2, blah, blah, blah. Those are all the names of this one protein, because different people discovered it and started studying it, and then realized later on that they were studying the same protein. And so you can see that they had to like just list all these names. And that's not very useful. So there actually was a group of people that came up with a standardization system for the proteins on the surface of leukocytes. Because you all could be working on different proteins on the surface of leukocytes. And you might want to figure out, am I working on the same protein as Tara, or am I working on a different protein as Tara? And start to figure out what's going on. And so a group of people got together. They get together every year-ish or so and actually do some standardization of the names of things. You can imagine it must be a real fun party. And they call these proteins parts of the CD system. CD officially stands for cluster of differentiation. That does not really matter. But basically, somebody picked one of the proteins and said, we're going to call this one number 1. And so they called it CD1. And then they said, this one we're going to call number 2. And so they called it CD2. And this one we're going to call number 3. So we called it CD3. And so in the back of every immunology textbook, somewhere in every immunology lab, all over the internet, you can just find tables of the CD proteins. And whenever people talk about immunology, they're like, oh my gosh, CD nomenclature is so confusing. It's just we gave numbers to all the proteins on the surface of the cell. That's all it is. That's it. So don't get freaked out. That's what this nomenclature system is. Yeah, Mark?
So who are these people? Is it like the immunology Illuminati?
It is it is a group of people who are particularly interested in nomenclature.
But like, how do they make sure it's standardized? Who is the governing body that's like, okay, we all
Yeah, I
do
was looking at that too. Like, okay,
I-
let's do that.
I don't remember the name of the governing body right now, but there is a group of people who go ahead and do this every year.
And then how does like everyone find out it's
They like publish it. Yeah.
you need to read this?
Well, basically, like when if I were to start working on a new protein, I would just like look up what we're calling it. Like, because they're probably like at least we're probably up to at least CD400 now, and so like most of them are already named, and you just like look up like what is the CD name for my protein.
I call 401.
You will see throughout the semester there are some times where the old name has kind of stuck. And so I'll be like, this protein is called blah, blah, blah. It's also CD86. But like, there are a few places where the old names have stuck, but there is anything that's on the surface of leukocytes, there's a CD name for. Okay, yes? Okay. So the leukocytes have all of these characteristic proteins on their cell surface. You'll see that many of them I have listed here with CD names, but not all of them, because like I said, sometimes the old names stick. And we just use antibodies against those proteins in order to distinguish our cells of interest. So if I wanted to detect CD8 T cells in the blood, I would use an antibody against CD3. That would bind to all T cells, both my CD4s and my CD8s. And I would use an antibody against CD8. And then I would look for cells that had both antibodies bound. As you can see here, I would generally want to have antibodies that had some sort of detection tag, like a red star or a blue star. And so I would look for the cells that had both reds and blues. If I found blue alone, that might be a CD4 T cell, for example. And so I would just do that type of detection. Yeah, Mark?
Why use both CD3 and CD8, not just CD8?
Because CD8's on some other cell types. This is dramatic oversimplification here. So I've told you that we're going to use antibodies in order to detect our cells. But I haven't told you what we're going to do to detect the antibodies, how we're going to find them. You could, if you wanted to, do immunostaining or lots of the other techniques that I already told you about. But let's imagine you did immunostaining and looked at your microscope. And let's imagine that you had to examine 100,000 cells in the microscope. How would you like to examine 100,000 cells in the microscope? You would not. Why not? It would be hard. It would take a long time. It would be tedious. So like I often in grad school would have samples where I wanted to look at least 100,000 cells for 50 samples. You can imagine this, that adds up real quick. And so there is actually a machine that does this in an automated fashion. That is sort of the workhorse of immunologists. And it is something known as a flow cytometer. With your flow cytometer, you take your cells and you first label them with antibodies or whatever fluorescent dyes or fluorescent stuff you want. And then you put them in the machine. And so here you can see that you have a part of the machine with cells that have already been labeled with antibodies. And those antibodies have stuff on their FC portion. Some cells didn't get any antibodies. Some got 1, some got 2, whatever. You put them in a huge volume of liquid. Here it's called the sheath fluid. Spoiler alert on our cytometer that we're going to use on Thursday in lab, the sheath fluid is this complicated stuff called water. You then can, with your settings on the machine, manipulate the way that the cells come out of this large area so that they are coming out in 1 cell per droplet of liquid. You do this, it depends a little bit on the type of machine that you have. This is depicting one that's dropping the cells by gravity flow. You do this largely by making sure you have a good single cell suspension and then playing with the pressure on the machine and to alter the speed at which droplets are coming out. I'm guessing your question is how you make sure, and there are some quality control things that we can talk about later. So we are basically going to have droplets with 1 cell per droplet coming out of this machine, falling. And they are then going to be hit by a laser. The laser is going to see a few different things. One thing that will happen is that cells that are different sizes will bend the laser light different amounts. So you can imagine this cell, the laser is going to shine on it, and you're going to get light go this far apart coming around it. And with this cell, you're going to get this going around it. And so you can actually get a measurement of the size of the cell based on how it scatters the laser. This is known as the forward scatter. It's a measurement of our cell's size. I'm going to explain the other one. I will tell you that I had a funny conversation where both Dr. Larson and I were explaining flow cytometry to our students at the same time. And her description of the other one was a little bit more physics correct than mine, which may not surprise you. So I'm going to give you like the simple version of the other one. Yes, Dr. Larson could explain it so much better than me. But we could also imagine that we might have 2 cells, and one of them might be a granulocyte. It might have a ton of stuff inside. And so if we have a cell with not much stuff inside, the light can go right through. If we have a cell with a lot of stuff inside, the light's going to bounce around a lot. And you can actually measure sort of the amount of bouncing around that did by something called the side scatter. Which measures either, you can either refer to it as the granularity of the cell or the internal complexity of the cell. You also may have fluorescent molecules attached to the Fc portion of the antibodies of interest. Your laser can excite those fluorescent molecules to emit light of a known wavelength. And so you can get, for example, if you use a 440 nanometer laser, you can get 480 nanometer light off of your this protein. And so you can actually look at the emission off of the dyes that are on the Fc portion of the antibody. And so I know, for example, that our cytometer that we have can detect light that's coming off at 522, I think that is, 564, 6 something, and 6 something else. Those are the 4 colors that our cytometer can actually detect. And so the cytometer will have a laser that's shining to the cell, and there will be the forward scatter detector, the side scatter detector, and detectors for different fluorescence emissions. The cytometer that we have has 4 colors in addition to forward and side scatter. So it has 6 total detectors. It's called a C6. There are cytometers. The one that I used in grad school had 21 colors. There are some that get to 50 or more colors. All of that data then goes out to a computer, and the cells go in the trash can. And so you end up with some data that looks like this. Yeah, Mark?
So for the ones that can detect 50 or more,
Yes.
what would be the point of something like that? Like to have so many different like fluorescent antibodies that are looking at different things.
So you might be looking at a very heterogeneous population of cells, and you might want to simultaneously look at different types of T cells and different types of B cells. You can also look at different molecules about the cell state. So what state of differentiation is it in? You can look at some of the signaling that's going on in the cells. And so you can get a lot of information about a lot of different cells. There also, when you start getting higher in the number of colors, will be situations where you don't use all of them, but you think about which ones work together the best. And so it's more giving you more options in terms of things like overlapping signals and things like that. So oftentimes, when you think about flow cytometry, you end up with data that looks something like this. This is a flow cytometry dot plot. And you will see that I have 2 axes with a whole bunch of dots. Each dot is a cell. And we are actually looking at one of the parameters, the number that the cell got, on one of those 6 parameters, either forward scatter, side scatter, or one of the 4 colors, versus one of the other ones. And with the software, you can kind of pick which 2 you want to look at against each other. So here we have forward scatter versus side scatter. You can also see the sort of number of cells that we're looking at here. When I was, again, as a grad student, I often would do 100,000 cells, and each sample would take about 2 minutes. Is what this is doing. And if you compare that to what you were doing with your microscope in lab 1, you can see how that's a big change. Our cytometer here, I don't run it that fast, but one could. There are other ways to detect the data. So you can look at contour plots that tell you about the density of dots in a particular location instead of showing you individual dots. There's also something called pseudocolor, where you get sort of coloring based on the density of dots, where you can see both the dots and the density. I point this out because in different papers that you see throughout the semester, you will see the data presented in different ways. But this is the same plot on these 3 different types of outputs. Yep.
So you said you don't run a hour cytometer that fast.
Yes.
What's the harm in running it faster or slower?
Then it clogs, and the last time it clogged, it didn't work for 9 months, and I had to pay $5,000 to fix it myself. I'm very touchy about that. So, and if you run it too slow, then you got to sit there for a long time.
Right.
You can also take that data and you can look at it in sort of 1 dimension instead of 2. So here I'm looking at 2 markers. Here I was looking at CD3 that had a fluorescent antibody on it. So how many cells had CD3 on their surface? And an antibody against CD8. So how many cells had CD8 on their surface? I can also do this as a histogram where I just look at the CD3 levels versus number of cells, or CD8 versus number of cells. And so we can get sort of that 1 dimension type of data instead of the 2 dimension. Every so often you're going to see histograms. But the real power of this is the other thing that you can do with the software. So if I have this plot, this is actually this plot again. If you look at this plot, you can kind of see that it looks like the cells are maybe in 3 groups. There's like a group that's here, a group that's here, and a group that's up here. When you see it in contour, you can actually see that a little bit better. There's really kind of 3 different groups. I can use my software to draw circles or polygons or whatever you want to call them around cells that I'm interested in. So I can draw 1 circle here and say, I like these guys, and 1 here and say, I like these, and 1 here and say, I like these. And those are called gates. And you can further analyze cells within a gate. So if you had 50 parameters, you could look at 2 here, and then you could double click on this and look at 2 more on just those cells, and then you could pick some, and then you could double click and look at 2 more, and you could sort of keep subdividing. You can also get numbers. So this shows that the 60.7% of cells are in this population. 3.81% of the cells were in this population, and 28.4 were in this population. This plot is a very standard plot of blood. You can see that these cells have kind of small-ish size. They're also really not granular. They're relatively small on the forward scatter axis, at least compared to these other 2. They're way at the bottom of the granularity axis. And cells with that profile are lymphocytes. So those are my lymphocytes, and I had 60% lymphocytes. These cells that have big size, kind of medium-ish granularity, those are the monocytes. And all these cells that have super granular are the granulocytes. And so, again, you can think about how challenging and how long it took you to do that in the lab. This is done almost automatically by this. Yeah, I had to draw the gates, but I've done it enough times that it doesn't really take much work. Yeah.
So I have 2 questions.
Yep.
First one is you would have to separate like white blood cells from red blood cells before doing this.
Yes.
And second question is why make the polygons so large? Why not like focus in on where it's like green or where it's red?
There are lots of sort of flow cytometry. That's like a flow cytometry person argument that we have all the time. And so that's one of those things we could talk about as thinking about how that would actually work on an individual experiment after class if you want. And so what I said is I could do my gating, I could make these 3 populations, and then I could further analyze each of them on, say, 2 other parameters, like CD8 versus CD3. And you can see that I get very different results from each of these 3 different populations. This is sort of showing you the same thing. I can gate this population and look at it again. I can look at my data as histograms. I can look at it as dot plots. And so I have a few different options. when I look at a plot like this, I might say, hey, look, it's kind of like there's 4 populations. Oftentimes, we're going to think about 4 populations, and we might even draw gates that look like a little cross. It's called quadrant gating. You don't have to draw quadrant gating. Quadrant gating is sort of going out of style now, but most of what I show you is going to be quadrant gating because it's easy. And I would say, oh my gosh, these are CD8 positive, CD8 high, because they're far along the CD8 axis, but they're CD3 negative or CD3 low, because they're low on the CD3 axis. These are CD8 positive and CD3 positive. These are CD3 positive and CD8 negative. These are CD3 negative and CD8 negative. And I could, if I had gates drawn on there, get percents of each of them and further analyze them and just go to town. So this is sort of what that data will look like. Often you might see it as a dot plot. You can see it with the gates drawn on it. And here you might get things like the percent of cells in this situation that are CD4 minus, CD8 minus is 3%. Here, about 5% of cells were CD8 plus, CD4 minus. 80% were CD4 plus, CD8 minus. So you could get that type of data. If I were to draw this on an exam, on a problem set, ask you to draw any of that good stuff, I would want you to draw axes and label them. You can draw the quadrant lines or not. Generally, you're going to be thinking about quadrants. And I want you to draw circles for the populations. And the size of the circle should indicate the percent. So there's 3% down here. That's a small percent, so I drew a small circle. There's 80% up here. That's a big percent. I'm going to draw a big circle. This one's like a little bit bigger. This one's even a little bit bigger. And so that would be what I would draw these data like on an exam, or what I would ask you to draw on an exam. So I would be caring about the size of the circle, big or small. Sometimes the size could change, and things like that. So this is actually how I got data like this for my dissertation. But I want to tell you one other thing, your fun immunology fact of the day, before I get going. Sometimes people also will use the term facts interchangeably with flow cytometry. If we are being extremely technical, they're not interchangeable. FACS stands for fluorescence activated cell sorting. When I showed you cytometry before, when we were done with the cells, they went in the trash can. There are specialized cytometers that can actually sort the cells for you and keep them. And so what will happen is you'll have everything that I've shown you before. The laser is going to hit the droplet full of cells, and then data is going to go to the computer. And the computer will say, oh, which one of these was that cell in? When the cell has dropped, the nozzle is going to actually put a charge on it. It's going to make it a positive or negatively charged drop. The computer will look, see what kind of cell it is, and it will either say, yes, I want this cell, or no, I do not want this cell. And it will flip the charges of plates and actually divert where that droplet falls so that you can actually sort out the cells based on the parameters that you set. So it's real-time sorting. You can then take the cells and use them for any type of experiment. So this is called a fluorescence activated cell sorter. These are way more expensive. But your fun immunology fact of the day is that this is known as a fax machine, FACS. It was patented in the very early 80s, late 70s. The year after it was patented, there was this other machine that got invented. And that machine made copies of things. That machine was called the facsimile machine. It made copies of stuff, and you sent them to people. And so they wanted to call it the fax machine. except that FACS fax was already taken. It had gotten patented the year before as the fluorescence activated cell sorter. The company who was making it was called Xerox, and they had X's in their name, and so they're like, hey, you know, it could be cool, why don't we do it with an X instead? And so they named it the fax machine. And so this machine is also the reason why the fax machine is called as a fax machine with an X. I will see you guys on Wednesday when we're going to talk about a paper.